Okay, so let's continue lecture 7, protein structure analysis. So one key aspect of a protein is to study its structure. And then we know the structure has 4 levels. The primary structure, that is a protein sequence, and the secondary structure, that could be helix, beta sheet, loop, and some others. And then they form this tertiary structure. Tertiary structure could have multiple domains. And then this quaternary structure could have multiple tertiary structures. That is, each chain form one part of the quaternary structure, and then they form a bigger complex. So by studying the structures, one can really understand the protein function very well. And also for other purposes like protein design, protein evolution, these structure provide a basis for such information. One step towards better understanding structure is the so-called supersecondary structure. We know we have the secondary structure, alpha helix, beta strand, but they form some kind of a special, we call it sometimes called a motif. There could be some simple combination of secondary structures, but many of these so-called supersecondary structures are highly conserved, which means that they occur very often in various protein structures. They often are part of a protein so-called architecture backbone, or they could be important functional sites. So let's see some of the supersecondary structures. So one common supersecondary structure is so-called helix loop helix motif. So here, from N terminals to C terminus, you have this helix, and then you have a turn, and then you have another helix. So this is actually the DNA binding motif. This actually is a calcium binding motif. So from helix here, and then have a turn here, and then has another helix. So you could have multiple conformations between the helices. It could be different angle, different length of loop, etc. One important one is actually so-called EF hand. This is a special helix turn helix motif. But in this case, you have a more clearly defined helices. Here, each helix is about 10 to 12 amino acids. And this loop actually has a fixed length. It has 12 amino acids. It has a very rigid structure, clearly defined. This is because you have a calcium that coordinate multiple residues. So this loop is not like many other loops, which are flexible. Here, this is a very rigid loop. And then this EF hand actually can form all kinds of structures. For example, this is a chopping C, and you see you have several EF hand structures. And then this actually can be an important architecture for its function. And then for beta and alpha, they can form, of course, a little bit bigger supersecondary structures. So we just talked about the 2 structure components. Now we see 3 structure components. It's the beta, alpha, beta. So you have 3 elements. And then you see these 2 beta strands. They are parallel. And then you see here you have a helix in between. So this is it can be represented like this. But if you look at the 3D view, this helix actually is above the plane of those 2 beta strands. So it has a special conformation. And this is also a cornerstone for a lot of different structures. One of them is called Rassmann fold. That is, you can have many of such units, beta, helix, beta, and continuously. And you can form such kind of a so-called Rassmann fold structure. And then we can see these 4 units forming some supersecondary structure. One of them actually is so-called is a 4 beta strand motifs. There are actually many ways to connect 4 beta strands. With some constraint, you have 24 possibilities, but not all possibilities are observed in nature. In fact, only 8 of them exist in known structures. Yeah, for example, there could be some very simple ones like here, this so-called Mindler structure. But this is not the most common structure, even though it is the simplest. The most common structure is so-called Greek key motif. So you have this 4 beta strand in this way, and then start from here. You have one so-called beta hairpin, and then you have another beta hairpin, and then connects to here. So this actually is important in terms of direction. So the starting point has to be this one. You can reverse the entire beta strand. That means you can go from here, but that conformation is not as common as this so-called Greek key motif. The Greek key motif, the starting one, is always the one in the middle, and go here, and come back here. Of course, yes. First of all, these are very similar to the Greek key motif. Yes, so this that's why it's called a Greek key motif, and it doesn't have to be in the plane. Here you see that it could be in this twisted format, and then it could form some kind of a barrel shape. So that basically shows the diversity of this supersecondary structure. And then supersecondary structure doesn't have to be contiguous in sequence. So the one we just showed is basically one contiguous strand. But some of the supersecondary structures are not contiguous. For example, this is a famous one called a losing zipper. So you have 2 helices, but they're not necessarily immediately connected. They can connect many other second structures and then come back, etc. So, so here, this basically not nearby in terms of sequence, but in structure, they are really locked very clearly, basically by these losings. So these losings were in between the 2 helices and pretty much locked the structure like this. And then you can see losing zipper can form all kinds of functions. One important function it forms is its transferrin factor. So this is losing zipper, and it basically attached to DNA and can regulate gene expression. And then the protein structure, you can also see the structure domain. So in a structure, typically you can see some kind of compact units. So we call it domains. Yeah, in fact, in evolution, these domains can be evolutionary units as well. So, so you can actually partition protein domains. Actually, we used to write software to do this kind of a protein domain partition, but nowadays it's pretty mature. In PDB, you can see the domain that is already partitioned. And then another thing that structure comparison. So structure actually is a much better conserved in evolution than sequences. So that's why many sequences, they don't look similar, but when you compare their protein structures, they actually can be very similar. One of the examples is the team Barrow. There are actually many diverse sequences that can fit into this beta barrel very well. And this actually is called a protein fold. That is it has a unique structure shape, it's compact, and it optimizes the packing of so-called hydrophobic residues to form a very robust core. So this is really driven by physical forces and also the solvent, etc. There are actually many folds discovered already. It depends on how you classify folds. There are some different ways to do it. But roughly, we're talking about a 1,000 fold discovered so far. Yeah, people estimate there should be upper limit of number of folds. But we, of course, we do not know. Some people say maybe thousands of folds. Some say it could be tens of thousands of folds. But comparing to possible sequences, it is definitely a much smaller number. Even comparing to sequence families, this is also much smaller. And then people classify proteins into a hierarchical fashion. And so you can see the protein sequences. If they group them together, then you can say this is a homologous family. These sequences or these proteins, they are evolutionarily related, and you can really see their evolutionary relationships through sequence comparison. And then different protein families, they can form a so-called superfamily. That is, you may not be able to see the sequence relationship clearly, but they could be so-called remote homolog. By using more advanced tools, you may be able to discover some of the sequence relationships. So in this case, all the proteins in a superfamily, they share a common evolutionary origin. Yeah, then going to an upper level, multiple superfamily can form a fold. That is the structure shape we talked about. And they may not be related in evolution. So sometimes two proteins happen to share the same structure fold, but not because of evolution. In evolution, this is so-called convergent evolution. That is, they came from different evolutionary origin, but then they happen to have some common features, in this case, a common structure shape. And then our upper level is a protein class, like this secondary structure, composition to all alpha helix protein, all beta, alpha plus, etc. So this is one common way people divide protein families. And this, of course, in this database called the SCOP. Yeah, SCOP actually is very good, but it's not frequently updated. So we will talk about another database called CAS. That is more up to date.